Sir, we are live now. Please start.
All right. Very good evening, everyone. All the students who are attending today's MOOC live session, the first live session for our two courses, Bioengineering and Interactomics. Welcome to this session today. I'm Sanjeeva Srivastava, your instructor for this course from Department of Biosciences and Bioengineering at IIT Bombay. Along with me, I have a set of TAs who are all going to be actively working with all of you over the next few weeks of this course. We thought it's a good idea for you to know us, that where we work, what we do, and also all of your TAs, because they will be interacting with you quite routinely. So today we are going to talk to you very informally, and also we want to talk in the context of at least a recent innovation which are happening in the omics technology in the context of pandemic COVID-19, right? So we just wanted to make sure that various technologies which you are going to study in bioengineering or in interactomics, many of those are quite relevant even in the context of COVID-19 and in the current pandemic which we have seen, that how these technologies can be very powerful. So this just overview slide, again, I'm not going to make today's session about another lecture. You're going to watch many lectures in the MOOC course, as you are already aware of. Each week you have 5 videos to watch, and you're going to do assignment from that. But I'm just kind of giving you the context of today's session, how we are going to plan it out. Our idea is to give you some glimpse of the research processes involved in doing this kind of innovation, a laboratory view directly from the lab, and also, like in the context, we'll talk about some basics of these technologies. Please do keep asking questions in the chat. As I get time in between, we will take your questions and discuss that as well. So COVID-19 is one of the recent pandemic which all of you have seen, and this is actually a good way to talk that how the current innovations and technologies in the multi-omics can be so powerful. When we heard about the Wuhan outbreak and the COVID-19, immediately there was how to detect the patients who are getting infected. So for something like as simple as that, you need detection of virus, you need to know the sequence of the virus, right? Otherwise, there are too many viruses and various type of similar strains are there. How to uniquely identify the new Wuhan strain? So idea was to first look at their genome sequence. And once you know that, within this genome sequence, there are some set of genes which are very unique sequences, can we now design the primers against those and come up with a simple test like the real-time PCR based test, right? And then along with that, various type of, electron microscopy experiments were performed, mass spectrometry experiments were performed to understand the proteins and the structure of this particular viral proteins. And then we knew about the spike protein, nucleoprotein replicases. And therefore, many type of tests and various type of possibilities towards vaccine development initiated, which got further characterized to look at the immune responses from microarrays. Various type of therapeutic strategies came forward. And throughout, we have seen like the need for bioinformatics and need for various type of, the computational power of analyzing the data, even tracking the viral strain moving to different parts in the same country and different parts of the world. All of that, all of you have witnessed that in, the entire textbook has almost opened up in front of you. So it's very relevant to recap that how these technologies have been very powerful at the time it was required. Of course, the virus has been much more powerful than these technologies and keep changing its, strain, and new variants are keep coming in, and we see second wave and third wave from Delta to Omicron. But nevertheless, I think you must say that human intelligence and the technologies have really tried to at least make a good balance of this fight, right, and this war. So with this context, today we are going to give you some glimpses and illustration of some of these technologies. Of course, your courses are going to give you a lot more fundamentals, but we are going to give you at least the overview of these technologies today. But before we move forward and take you to the lab and do some of these experiments, let me start by introducing you to some TAs who are actually very well qualified PhD students from IIT Bombay and some from IIT Madras who are going to be with you in the course. And I'm going to introduce to them one by one, so that you are aware that, to whom you're going to talk in the next couple of weeks' time. So let me just stop sharing my video, and I'll introduce you to subsequently in the follow-up sessions with the different TAs.
Hi all, my name is Shalini and I'm a PhD scholar at IIT Bombay under Professor Sanjeeva's guidance. My work revolves around the understanding of pathogenesis in Plasmodium vivax of Indian population. So my work is majorly divided into two parts. One is to understand the disease, where we take the whole blood sample and we try to understand the multiple OMICS studies, that is metabolome, proteome, and transcriptome of the plasma samples and the parasite. Then we try to utilize integrated omics to compile these studies together to understand how the disease is progressing. The second part of my work also involves the comprehensive search or exploration of the biomarker candidates for diagnosis and prognosis. For diagnostics, we have taken the parasite proteins, where we have tried to express and do the immunoassays to prepare a multiplex kit where we can diagnose the parasite using the parasite proteins and prognose the disease by using the host system in the clinical settings. So once that goes in the clinical setting, it will have more accurate diagnosis, and that can be further used for understanding the disease progression better. So this is about my work, and I have an inclination towards this course because I feel that as we move on, we forget to see the basic things with a different perspective. And with this course helps me to connect to the people who are starting in this field from the very beginning. That gives me a whole different perspective. This fascinates me and helps me to understand or to look at a particular thing with different perspectives. So I look forward to interact with you all more and to learn with you. So welcome, guys.
All right. Thank you, Shalini. Now let's move on to next TA, which is Amrita.
Hello, everyone. My name is Amrita Mukherjee, and I'm a PhD student at IIT Bombay, currently working with Professor Srivastava. My work focuses on understanding the biomolecular signatures of cervical cancer. It's a small part of Cancer Moonshot program as well. So I perform proteomics and genomics from human cervical tissue, both cancer and tumor, and also on different cell line models as well. So for the same purpose, I'm also involved in developing a drug-resistant cell line models of cervical cancer. By the way, I'm currently the TA of the Bioengineering course. So this is the third time I'm being a part of this MOOC course. So previously I was involved with the same course twice. So one of the reasons to be in the MOOC course as a TA is that I'm very much fond of interacting with students and love to share and discuss ideas. And as you all know, MOOC is a huge platform that brings together various types of participants, which are the blooming minds from every corner of this country. So it is a great opportunity to explore the connectivity as a teaching assistant here. So in this course, I'll be helping you with the contents of week 1, 6, 7, and 8, which includes the basic biology for engineers, cell cycle dysregulation and cancer, proteins and proteomics, as well as bioinformatics. So I'm very much looking forward to interact with all of you. Have a great day.
Thank you, Amrita. And we are also introducing this time a new scheme in which the Prime Minister Research Fellowship recipients, now Lavanya is going to talk about her work from IT Madras, is going to give you a help session towards your assignments, right? So let me introduce Lavanya now.
Hello, everybody. I'm Lavanya. I'm a PhD scholar in Protein Biophysics Laboratory, working under the guidance of Dr. Adi Narayanan at IIT Madras. My work focuses on the study of antibiotic binding proteins. Specifically, we are interested in mirror family of proteins. So these proteins bind and capture antibiotics and prevent them from acting on the bacterial cells. Therefore, mirror family of proteins help bacteria to gain drug resistance. So the gene encoding for this protein is cloned into a plasmid vector, which is then transformed into E. coli cells. The cells are cultured and the protein is then purified through a series of steps in chromatography, and the purity is checked through SDS page, polyacrylamide gel electrophoresis. The purified protein is then characterized through various techniques such as circular dichroism to see the secondary structure motifs, and other techniques such as differential scanning calorimetry, intrinsic titration calorimetry, SACS, NMR, and intrinsic fluorescence. So through these characterization techniques, the structure and folding of the protein is understood better. And we hope to have some applications through these in the field of medicine in generating new and effective drugs that will not bind to the antibiotic binding proteins. So this will help us get a better understanding of folding and binding mechanisms in multi-drug resistant systems. So I'm one of the TAs for the course Bioengineering, an interface with biology and medicine. So this is a beautiful course that connects the science in biology with cutting edge technology in the field of medicine. So this is my first MOOC course as a TA, so I'm equally excited in being a part of this learning experience. In this course, I'll be helping you solve the assignment questions by giving you some practice questions, and we'll be solving it together in live sessions that I'll be conducting every Friday evening 6 to 7 p.m. I will also help you clear any doubt that you have during the course.
Thank you, Lavanya. And next, let me introduce you to Ankit now. He's another PMR student.
My name is Ankit and I am a PhD scholar at IIT Bombay working under Professor Sanjeeva. My work is based on meningioma tumors. Meningioma are a kind of prominent brain tumors, but they remain mostly benign in nature. But only a fraction of the patients generate aggressive or grade 3 kind of tumors. So my work focuses on identifying the biomarkers that can be easily help in early onset of detection, and also to develop mass spectrometry based immunoassays so that the patients can be assessed easily using body fluids such as serum. Also, I am looking on different biophysical characteristics that will be helping in stratifying the grade-wise classification of the patients so that we can get an idea of which patients are developing the aggressiveness and can try to prevent them. Then we'll perform an integrated OMICS analysis to identify the altered and the most perturbed pathways that are involved in meningioma progression. Finally, I would like to identify one drug candidate and perform drug repurposing based approaches using both in silico as well as cell line based assays. I am doing TAship for the MOOC course for the first time. I will be conducting a hands-on bioinformatics session every weekly to give you all a glimpse of to get an idea of all the data that is generated through different technologies that you will learn in both the bioengineering and the interactomics courses, how to analyze and interpret those data. So I took up this TAship for this conducting this session to interact with you all intellectually as well as to learn from you all. So it will be an interactive session and a hands-on session that will hope to interact with you all soon. Thank you.
All right, thank you, Ankit. And now let's move to TAs of Interactomics course, and let me introduce you to Arvya Banerjee.
Hi all, welcome to this Interactomics course. My name is Srivastava and I am a PhD under the mentorship of Professor Sanjeeva Srivastava. My work basically focuses on the pituitary adenomas. I am the TA for the Interactomics course. I have around 5 years of teaching. The reason for being a MOOC TA, I like to having intellectual interactions between the participants and also to self-educate myself. In course, I will be helping you with the protein microarrays section, how the protein microarrays helps in the diagnosis, how they are fabricated, and what is their use. And I'm very positively looking to interact with you all. So welcome to this pituitary research, and I am performing this research under Professor Sanjeeva Srivastava. So first of all, our aim was to build the detailed proteome map, like the blueprint or the landscape or the anterior and the posterior lobe, because till date there is no proteome available for the anterior and the posterior proteome. So we have performed the deep mass spectrometric experiments to decode the proteomics of the both anterior and the posterior lobe, where in this pituitary proteome map, we looked at the missing proteins, the dark proteome, the UP proteins, the UP proteins, and the proteins that shared biological functions and which help in the locomotion. Second is we are studying the Cushing disease. The Cushing disease and Cushing acromegaly and NFP are the kinds of the pituitary adenomas. In the Cushing disease, we compared the Cushing disease with the normal controls, and we wanted to study how these tumors differentiate from the normal controls and the protein human locomotion. In the acromegaly is also one of the rarest control where you will find the gigantism, atrophy, enlarged feet, cardiomyopathy, hypertension, this kind of comorbidities. In the acromegaly, we also compared those with the normal controls, and we wanted to differentiate how the proteins behave differently in case of the acromegaly. Lastly, the NFPA, adenomas, we wanted to see like how these non-functioning pituitary adenomas are different from the normal controls and how we can differentiate those. Moving to the grade-wise classification, the pituitary adenomas can also be classified on the basis of the grade by the position in the cavernous sinus and in the sella tunica. So it can be defined, it can be designated as the grade 1, grade 2, and the grade 3, depending upon their size. And basically what we did there, we compared the different grades with the normal control and performed the proteomics to know how different grades can be correlated with the protein markers and protein related to its biological functions and human locomotion. Lastly, we also studied the metabolomic profiling, where we detected the autoantibodies, like which are the autoantibodies which can be diagnosed at a very early stage using the serum sample. And also we performed the FDIR and metabolomics of the serum samples of the pituitary to encode the metabolome change that was very much responsible for the project. So this is my project introduction, like what I am working on. I will very happy to take questions. Thank you.
All right, thank you, Arghya. Now let's have the second TA for Interactomics course, Abhilash.
Hi all, I'm Abhilash. I'm a PhD student in Proteomics Lab, IIT Bombay. I'm working under Professor Sanjeeva Srivastava, and my work focuses on colorectal cancer proteomics for identification of biomarker that can detect the cancer at a very early onset. We focuses on different multi-omic data for the discovery of biomarkers. After the discovery, we also try to take the biomarker for validation using molecular and cellular proteomics approaches. This will tell the role of the marker in the prognosis and diagnosis of the cancer, as well as other in-depth biology. After inhibition of the marker, if it is upregulated, what happens to the systemic effect? So this work we do using molecular cell culture approaches that will tell the role of the protein marker in pathogenesis, development, or metastasis of the cancer.
Thank you, Abhilash. Let's now move on to last TA, Avinash.
My name is Avinash Singh, and I'm a PhD scholar at IIT Bombay, working under the guidance of Professor Sanjeeva Srivastava. My research work basically focuses on the multi-omics exploration of oral cancer. So oral cancer is one of the common type of cancer in India. So in this study, I'm dealing with the proteomics, genomics, transcriptomics, and metabolomics study of oral cancer tissues, saliva, and serum. And after that, try to find a potential biomarker and different therapeutic targets for the oral cancer. I am a TA of the Interactomics course, and I have 1 year of a TA assistant experience in IT different courses at IIT Bombay.
And the reason to be in a MOOC course, MOOC course is to have an interaction of all the participants coming from the different parts of India and different background. So in this course, I will be helping with you PLI and FDIR and the role of application of this instrument. So thank you. I'm looking forward to have a positive interaction with you all.
All right. Well, so we have seen, like, we have several who are going to help you to be part of this particular course. I'm just going to take you to the lab. But before that, can I request all the TAs to just mute themselves? A lot of backgrounds are coming. Flash and others. No. All right, I hope that's better now. So this year we are just, on the experimental basis, we are trying to add 2 more activities for our MOOC course. We always want to make it more and more interactive. So this year we are going to have Lavanya with you every Friday, especially for the bioengineering course, right? And she's going to solve the bioengineering assignments of previous year to give you some idea. And then she can take some query for the current year questions as well. But of course, she's not going to help you solve your own assignment. You have to do that. Additionally, Ankit is going to do the bioinformatics session, which will be a kind of, optional activity for you to learn more. We do see that, the way data analysis has become very powerful and very important. It's a good idea for you to be aware of the basic tool for bioinformatics, and also, like, in terms of how to go take your knowledge forward by doing something available from the internet tools. So it's not going to be for your exam, but essentially towards your own learning. Both for bioengineering and Interactomics course, we are going to share the Google form so that you can take the slot, and every week we will have this session to give you more hands-on activity about doing certain type of analysis. Sometimes it will be basic bioinformatics, and sometimes it may go towards some genomics and proteomics and various type of advanced data analysis as well, right? So these will be some new addition to our ongoing activity. So you have seen for both the courses, for the bioengineering and Interactomics, we have a very well prepared team, and who is quite already experienced in this area. These are all very good PhD students from different courses at IIT Bombay, and they will be with you to solve your queries and problems. So I think you should make this as an opportunity to interact more with us, and don't lose that opportunity. Just being it is an online course, we do not want you to feel that we are only online. We are definitely available to you like a classroom, and just make sure that you are taking that first step forward to interact more with us, right? All right, so in this direction, let me just now not talk, because it looks like we're just talking. Let me take you to the video. I just took my lab coat, and I'm just going to move to the lab to show you where we work, right? Just briefly. And of course, as we go along in the subsequent live sessions, you will have more interaction on different technologies. So I'll just kind of walk around. So this is our proteomics lab at IIT Bombay. Various type of instruments and molecular biology tools which you learned in the courses are all available, starting from something as basic as PCR, which will be explained to you shortly, towards various type of even high throughput approaches, including the technology like protein microarrays, which something you can see it here. And of course, you need to see a lot of the biological samples, how they are being stored, how they are utilized. Lot of temperature control systems and filters are required for that activity. We also have various type of sample preparation workstations, which is very much required for doing your sample prep, especially if you are looking at thousands of genes and proteins. You need much more sophisticated these kind of instruments and technologies. One instrument that you see here is FDIR. So that is another instrument which will be talked shortly. But I'm just moving around to give you the glimpse. Of course, more and more of these things will be explained to you shortly. We specialize mainly in the technology known as proteomics, which is a bunch of technology to study the proteins in more high throughput manner. So my lab has more specialization in this particular area. And here you see a lot of very high advanced gadgets which are available. So let me just stop sharing and see if probably you can get a bigger view of the lab. So this field of proteomics, has really advanced further to genomics. And now goal is to look at all the proteins, how they interact, where they're localized, how they function, can we understand their modification. So various type of properties, very challenging to study for protein. And studying at the scale of thousands is really makes it difficult, because if some of you would have done a short project or some internship somewhere to look at one protein structure or to purify one protein and to look at protein purification, you must have encountered a lot of challenges, right? So studying thousands seems very challenging. But these are somehow the very state-of-the-art technologies like mass spectrometers, which has made it much easier for us to understand and study these fields. So we will be talking about these type of instruments. What you see here is the liquid chromatography to separate the peptides, and then the mass spectrometers, which can separate these ionized peptides based on the mass and charge ratio. And you can then look at the databases to analyze that data set. Another type of instrument, another mass spectrometer, could be utilized for metabolomics analysis, looking at the small molecule and drugs and metabolites in our given sample. Or instrument like what you see in my back is known as a triple quadruple mass spec, can be utilized to look at specific targets, a specific metabolite or a specific type of peptides, which could be analyzed. Of course, we will be talking about NGS, which is very important next generation sequencer instrument, which is has given so much idea during the pandemic time, right? And if you want to study some of the interaction in the high throughput manner, how a protein is binding to the drugs, if you want to test out, for the spike protein and it's going to bind to which drugs, you need more technologies, and therefore techniques like biomolecular interferometry based platform Octet, what we have, or surface carbon resonance, be a core instrument, some of these technologies can be used. So I'm sure you now feel excited that we are going to talk about many recent technologies which is available for us to introduce to you, which you will be talking and discussing in this course at great length. But we have pretty much all of these gadgets and these instruments in our lab, and we are going to talk more and more about them as we go along. So give me a second to just, take off my lab coat, and I'll be with you to conduct the session now. All right. So I've not seen any query. Probably, I'm not sure if I'm missing, but let me welcome all of you once more, those who are joining a little late, and let me just name some of you. So Pooja, Supriya, Anisha, Sayan, Naushad, Manoj, Gharshita, Mumtaj. Hello, everyone. I think all of you have been posting some messages on the chat. So hello and welcome to this particular session. We are available to talk to you in today's live session. What we have planned, that we are going to give you some story and the glimpse of technologies in the context of COVID-19, right? Which is an invisible enemy in front of all of us. It still keeps surprising us with various type of challenges. And during this type of, the course of pandemic, we also did a lot of research, and we are just trying to show you in the context of some of these instruments and technologies, how it can be, used to, advance our knowledge, right? So toward this slide, we started thinking about, there are many detection tests available, but also those detection tests, which is conventional like real-time PCR, requires the RNA from the virus to be amplified with the cDNA to be converted, and that has to be amplified with the real-time PCR. And that's where, and you are looking at specific primers, and that's where you see a lot of misdiagnosis also happening. So idea was, can we look at directly the peptides of these viruses, which is given that the spike protein is quite in abundance, so where the nucleoprotein and the replicases, can we look at directly the peptides and proteins from the virus rather than amplifying the RNA and converting those to cDNA? So that was one question we wanted to address. And second was, can we look at how a human changes in response, host response because of viral infection, as the disease gets progressed from the mild to the severe form. that is known as prognostic marker, right? And in this case, we have various collaborators, various clinicians from different hospitals, Dr. Jayanti Shastri from Kasturba Hospital, Dr. Sachi, Dr. Om Srivastava from Jaslok Hospital, Professor Sanjeev Sabnis, Maths Statistics Department, IT Bombay, Professor Michelle Hill from Australia, QMR. Many collaborators and clinicians were involved in helping us doing this very challenging project forward, and some glimpses gives you idea that how difficult it was to do this kind of research, especially in the first wave of the COVID-19 pandemic, when it was very challenging to even think about handling the COVID samples. So what we thought, can we take the patient's nasopharyngeal swab, which is already being tested for the real-time PCR test, and from that swab sample, can we get the peptides out by doing different type of extraction methods, use the mass spectrometry to look at which peptides we can detect, and we could see many peptides on the virus we were able to find out. And then can we specifically detect those viral peptides using a triple quadruple mass spectrometry based method, which Avinash is going to talk to you in subsequent session. And then based on those, we were able to segregate negative and positive, and as well as some patients who were recovered, but is still showing the trace response of the viruses. So this was one of the things which we thought to develop. And further, so this work actually was one of its novel contribution, which was also we got invitation from Nature to give a talk globally to tell about these technologies what we are developing. Additionally, we thought, can we look at patient prognosis, how the patient is moving from the mild form to the severe form. And if you imagine, everybody who gets COVID, becomes apprehensive, like, am I just going to recover, or am I going to get deteriorated? Whether I'm going to need the ICU, or I'm going to just be fine in like any other viral infection, right? So that's always that challenge is there. And therefore, can we find the cues or the mechanism or some sort of indication in the body which tells us, okay, there are set of protein which is now deteriorating or they are changing, which is giving us some clue that patient is now going to go, deteriorate. Whereas if it is not changing, it may indicate this patient is going to get stable and getting recovered fast. So there are some tests which you might have heard. Doctors were also looking at some of these type of tests like D-dimer test and looking at IL-6 level, ferritin. Some of these proteins you might have heard the names, which were actually indirectly helping clinicians to find out whether they should take the patient for enrollment or they should be just doing it fine and they should release. But what we did, we look at very systematically almost 200 patients blood sample using these type of technologies from both mass spectrometry as well as FDIR. And then what you see here, a kind of a test, I'm not going to talk in great detail here, but it shows that from the non-severe to the severe form, set of proteins are very different, like angiotensinogen, apolipo-B protein, serpin protein, fibrinogen gamma chain protein. So some of these proteins, like a set of protein, when they are showing indication that they are very different and their levels are very high in the patient, that could be indicative that patient is might getting severe and prone to severity. So that's where I think this technology and test was quite prominent, and we got patent and a lot of leads from this particular work, which was also highlighted at the cover page of the Journal of Proteome Research publication. Further, we thought, since there is a lot of change happening in the plasma sample, can we come up with a quicker test and quick way of detecting those signals? While mass spec is very authentic to give you the exact sequence, exact protein, but can we use other technologies which could be at least indicative, but very quickly at the point of care devices? And that's where we use this type of carry instrument for the FDIR, which was from Agilent Technologies and became very powerful for us to find some very unique signatures, which is indicative from the differences of non-severe and severe group of patients. Again, Argu is going to talk about this. This work again got covered in the ACS journal and a lot of media highlights. So with this, I think, you got some glimpse that the technologies which you're going to study in this course are very relevant, and they're as relevant that they could be utilized at the time when we need them, right? And therefore, it's really important for you to start knowing the fundamentals and seeing the broader perspective where it can be used. So we are going to now give you some live demonstrations from the lab, one by one for different techniques. And, broadly, of course, the time is going to get short to give you a very detailed presentation for each one of these. But I'll request TS to just quickly give you the glimpse of those instruments and their principle. And as we go along, then, of course, we will talk more to you about each one of those in detail, right? So let me invite Amrita now to talk to you about how PCR can be used and real-time PCR can be used for broadly for any disease for that matter, and especially for COVID-19.
Hello, sir. Thank you. Thank you for good evening, everyone. So I'm going to talk about the PCR or polymerase chain reaction and the modification of PCR that has been really helpful in this COVID scenario. So as you know, the PCR or polymerase chain reaction, so it is a simple, it's not as simple, but it converts very small amount of DNA into, it amplifies very small amount of DNA into very large numbers of billions of strands of DNA or target DNA template in very small time. So basically the amplification reaction is composed of three steps. Step 1, 2, and 3, as mentioned in the slide. So the first step is denaturing. So that before going to the step, I would like to know that we need different starting materials. We need the template DNA. So the template DNA you are going to amplify. Then we need some primers. The primers are forward primer and the reverse primer. So these primers actually decide up to which section you are going to amplify in your template DNA. And then we require a special type of DNA polymerase enzyme as well. So this DNA polymerase enzyme is not the normal DNA polymerase enzyme. It's a enzyme that works at a very high temperature. So it is isolated from the TAC thermosupportic organism. So it's called a TAC DNA polymerase. So the step 1 is denaturing, where the two strands of DNA at the higher temperature, like 95 degrees centigrade, they separate. And then then comes the annealing step, where at around 55 degree to 60 degrees Celsius temperature, both of the primers bind to the both of the strands of the DNA. And then at around 72 degrees Celsius, you allow both the primers to synthesize the DNA. Now, these three steps are repeated multiple times to amplify the entire reaction, and at the end, you will get billions of strands of your target DNA from the very small starting material. Now, how this PCR techniques has been useful in COVID-19 research. So, a small modification of this.
Could you please turn on the video and also keep showing the instrument briefly, at least thermal cycler, and
sure.
Then continue. Yeah.
Sure, sir. So, sir, if you can highlight me, so I'll be able to.
Right. Yeah. Please continue. Yeah.
So let me know the machine is visible or not.
It is, Amrita, but PPT is actually come on the full screen, so we cannot highlight on the full screen. You have to continue.
Okay.
Yeah.
Oh. Okay, right. It is visible. So this is the machine we have. This is the instrument. We call it a thermal cycler. We can run the PCR experiment in this instrument. So this is the lid of the instrument. You can open the lid and you can place your reaction mixtures here. Now, after placing the reaction mixtures, you need to set the parameters. So this is the pop-up window where you basically set the parameters for running the experiment. So I'll start by clicking with the new protocol, if you can see. So if I start with the new protocol, so a window will appear where we get to choose a lot of options from there. So starting with the very first lane, the very first lane is a section where we allow the samples to preheat. So we have already told that the PCR is composed of 3 cycles. One is an separation of strands. So before separation of strands, we preheat the samples at 95 degrees Celsius. Here you can actually set the timing for the preheating. We have, for now, we have set it 3 minutes. You can change it accordingly. Then the step is, hello? Okay, so the next step is the denaturation step. The denaturation is always performed at around a very higher temperature. Here it is set as 95 degrees Celsius. You can set the timing accordingly. After denaturation, the next is the annealing. So annealing we have set here at the 55 degrees centigrade. This annealing depends. Annealing temperature depends on the melting temperature of the primers you are using.
Yeah, sorry to interrupt. But I think looks like we are getting late now. So what looks it is I'm sure everybody got to feel that the same principle which you showed, those settings has to be made on the instrument. You can just simply open once more the lid of the thermal cycler I have highlighted now. So here, as the students can see, like in the 96 well plate format, you can actually do a lot of reactions of amplification of DNA simultaneously, right? So once you have prepared your sample, you can also optimize multiple conditions for different type of primer and even different temperature controlled sensors could be there when you can optimize multiple condition, or you can do many sample at the same time. So all of this is very high throughput. And now Amrita will just recap quickly what all of you already know about how the real-time PCR has been used for testing the SARS-CoV-2, right? So Amrita, just walk them through this slide now.
Right. So in case of real-time PCR for detecting the SARS-CoV-2, what we first do is that the swab samples are collected from the suspected patients. After collecting the samples are first inactivated either by heat inactivation or by chemical inactivation. And from there, the RNA of the virus is extracted. Now, we know that the PCR reaction happens with static material of DNA, but in the modification of this such PCR, so the viral RNA is first converted to a complementary DNA with the help of a special type of enzyme, which is reverse transcriptase enzyme. And then this complementary DNA is getting amplified to finally produce a number of, with the help of the SARS-CoV-2 specific primers, to finally get the ultimate numbers of amplified DNA. So, yeah, here in this slide, you can see the mechanism of the principle of reverse transcriptase PCR. So viral DNA is there, and from there you extract the RNA, and the entire process is being performed. So so that's it.
Right. So yeah, I think thanks, Amrita. So again, students, you can appreciate that something as simple as PCR and real-time PCR, which is pretty much available in all the standard molecular biology labs, became so powerful and useful to detect this particular virus SARS-CoV-2 at the time when we really needed that, right? And that's where I think these technologies have made their impact so much. But how these sequences, we knew about them. So that actually comes with the next generation sequencer. And NGS became very powerful because initially we have no idea that what this strain is infecting us. So how to develop the test, right? So now I request Ayushi to just show you our NGS setup, and then she can briefly talk about that.
Yes, sir. Okay. So I'll start the video. So hello everyone. We have in our lab we have Ion Torrent S5 Studio, which is a sequencer here, and it comes with other small instrument which helps us in the library preparation. So this instrument basically is very good for the primer-based targeted sequencing, where you have the primers of the sequence that you want to amplify and then further sequence. So first step. So there are 3 steps in the sequencing reaction. So the first step is the library preparation, where you add the primers and the reaction mixture for the library preparation, and then you add the adapters and barcodes so that you have the library prepared. And the second step is the template preparation and amplification. So this instrument is called Ion One Touch. This particular instrument works on the amplification of the library that you have prepared. So here this is the adapter where you fill the reaction mixture, and this will help in the clonal amplification of the library. And here we place the 2 recovery tubes, where after the clonal amplification you get your amplified library in the recovery tubes here. And once you have your template positive library, you can then go ahead with the sequencing reaction. So for that, so the instrument has this chip So here you can see in the one of the oval structure, we actually load the sample. So this chip has multiple small wells. Each well has the sensor attached to it. And when you load the sample, so you must be familiar with the when the phosphodiester bond form during the DNA formation, there's a release of proton ions. So this actually the well, which is where you load the sample. So this well has the pH sensor attached. So whenever there's a release of proton ion, this pH sensor will actually detect the proton ion and give the instrument the signal that the sequencing is happening. And here, this chip will actually go here in this instrument like this, and then you can, you can just place, close the chip clamp, and you can fit the, this is basically the cartridges which supply the 4 nucleotides. You can place the cartridge here like this, and this is just the washing solution and the cleaning solution. You have to close the sequencer, and then you can start the sequencing reaction. Right. And
Great.
now coming back to the
So there are many principles, right? As Aishi, you are just mentioning one of them with the ion tolerant technology, looking at hydrogen ion detection. But there are many principles involved. Different type of next generation sequencers are available. We are not just emphasizing about one technology. We are just giving you the glimpse, all the students, just a glimpse of one type of NGS technology. That's how sequencing could be performed. So therefore, we will not spend a whole lot of time. But what you can see, theoretical concept, what is shown on the slide, how practically is very easy to do that actually inside the lab. And with these kind of instrument and technologies, the time when we really needed that, we quickly got the genome sequences of this. When then after that, with the basic bioinformatics sequence alignment, it was found it is not forming very, it is coming to the coronavirus family, but it's still a novel variant, novel strain. And therefore, then it was named the COVID-19, right? So different steps involved in doing the NGS. I'm sure in the theoretical lectures, you will learn more about it. Aishi, I think given the short time what we have available, probably we'll skip this slide now. But students have at least got the glimpse of the instrument, which is quite advanced instrument, but it is very simple to perform these experiments. Concepts are very important and novel, which is what all of you have to pick up at the basic level. So once we got these NGS instrument and real-time PCR to do the detection, of course, then many type of, whole world started reporting about the COVID-19. Everywhere is spreading, and, all kind of international flights got banned, and everything what you have seen in front of you were result of these technologies, right? But then idea came that can we look into some other type of ways of quick detection, various type of antigen test, antibody test, immunogenic various type of assays. All that started coming up because the problem was spreading so far rapidly, the whole virus everywhere, that it was very difficult to rely only on one technology. So therefore, there was need to look into multiple other alternative platforms. And now Argu is going to talk about spectroscopy-based applications for COVID-19. All right, Argu, over to you. Please unmute and start.
Yeah, okay. Am I visible? So hello everyone. So I am Orgo. I will be showing you the FDIR instrument. So what instrument you are seeing? This one. So what the instrument you are seeing is the FDIR instrument, the cutting edge technology. It is very small in size. You just take 1 micromilliliter of the sample and you put it into the diamond slit, what you are seeing here, and the infrared. And the infrared can be easily visible, like the infrared means there is a mirror and the infrared spectrum is recorded. Now what happens, you can directly take a minimal amount of serum samples directly. Now these serum samples will be containing the metabolites. These metabolites will show vibration, and that vibration upon the FDIR will be recorded here. So here you can see the peaks, what are the formation, and through these peaks we have to decode like which is COVID positive, which is COVID negative. And in COVID positive also you can decode like which is severe and which is non-severe with the help of ML based application. So this kind of vibrational spectra is good enough to distinguish the metabolites and characterize as per their severity. Back to you, Professor.
Very good, Argo. So I think this work which Argo and team did was published in Analytical Chemistry and got quite a bit highlights and became a very easy way of detection of severity in the COVID-19 patients. So moving forward, I think another mass spectrometry based technology, which you will be also studying quite a bit in this course, a new advancement based on the mass spectrometers are known as targeted proteomics. And that's where different type of clinical assays being developed, which is like a substitute of ELISA, right? Like the way in the clinics ELISA happens with antibody-based testing, without having availability of antibody, could we still do the test? And that's where I think, idea was to look into targeted proteomics-based assays. So Avinash is going to show you the triple quad mass spectrometer and how these assays could be developed to specifically detect the viral detection as well as prognosis of the patients.
So, hello students. So basically, this is the TSQ voltage instrument, and this is the SPLC. So this instrument is basically it can show.
I cannot see that. Yeah, now we can see that. Please continue.
Let's turn on.
Please go ahead.
Yeah, okay. So as you can see that this is the TSQ voltage mass instrument. So this have a basically mass analyzer triple quadrupole, which can be used as a different kind of study like a pharmacological studies and the clinical research, as well in the forensic toxicology to for the doping test as well. So it can detect a very small amount of very small molecules as well as the peptide by study. So this is it is connected to the UHPLC. So this is the HPLC basically where we keep our samples. So here you can see that. So here we keep our samples, and after that, this samples is the passes through the by column to the instrument, and where we this is the ion source where the ionization of the molecules or peptides happens. After that, we detect the different type of targeted peptides. So as you in slide and also in the screen, you can see that. So here is the representative diagram of a few peaks which have isolated after the discovery study, and that after that we have identified few proteins which are basically differently expressed in the COVID positive and COVID negative. So here you can see that there is a first upper three samples are basically COVID positive, and the lower three samples are the COVID negative samples. So these peptides are basically upregulated in the positive samples and downregulated in the negative samples. So similarly, these are the different peptides and proteins of so as you can see, there is a different peptide we got from the study. And also in the slide, you can see we have also published a few paper on the basically the COVID plasma sample as well as swab sample. So yeah, so this is a very good instrument to detect a very small amount of peptides and also the small molecule as well.
Great. All right. So this instrument and some of the other mass spectrometers we are going to cover in a dedicated session to all the participants. This is one of the cutting edge technology, not only for COVID-19, but for cancer research or drug discovery process is very important to move forward in this area. Another technology which is quite a bit being used is the microarrays. From the genomic-based microarrays to the protein-based arrays, it has a lot of utility. And especially in COVID-19 context, it was important to look at immunogenic responses. And in many kind of cancer, one is looking at early detection of the biomarkers. So again, protein microarrays become important. So Abhilash is going to now walk you through with this microarray-based workflow. So while the TLs are getting ready, here what you are seeing is protein arrays, which is having a small on the glass slide. You have thousands of proteins printed, and the principle is very simple, like a Western blot. When you have we are looking at antigen antibody interaction, right? So here in the same way, like the antigens are printed on the chip, and what we are showing that if a patient sample comes in, which let's say a serum or plasma sample, whether that is having antibody in response to disease, and that antibody is going to bind to the antigen on the chip. After subsequent washing step, then you are going to label with fluorophore, which could be used to detect that signal. So now I think you can see on the screen here, which is one of the microarray slide here, which is being shown, where you can see these are the very high density arrays, and the chips where you have thousands of protein printed. In case of human protein arrays, we had almost 19,000 human protein in duplicate printed. And in case of the SARS-CoV-2, we had all the peptides of the various protein of virus were all printed. So again, it's very high density format to look at the entire proteome at a single snapshot. Look at the immunogenic response using different type of code which you can use to detect the signal. So thank you, Arko and Abhilash. And let me now move on to the next technology, which is the looking at biomolecular interactions. And that's where you can think about how to use the protein-protein interaction or protein-drug interactions. And now Shalini will walk you through the BLI, Biomolecular Interferometry based principle.
So, hi all. I hope my video is visible.
Shalini, please continue.
Hello.
Show BLI instrument and talk.
Is this okay? So, this is the BLI system. There you can see there are these two things. One is the tray, which is containing the sensor, and the other tray is there, which is containing the sample. This is the samples, and these are the sensors. So here, if you see, in the sensors, we know that these biolayer interferometry is actually having first step, which is containing all the different types of chemistry which is involved before understanding the kinetics or protein-protein interaction or different molecular interactions. So what we do usually, these two are the places. One is for the sensor, where we place this kit, and this is exactly 96 well-placed replication, which is done here on the black plate of 96 places. So here all the samples will be kept and all the buffers will be placed, and this template which you can see here are the 8 sections which pick up the 8 sensors from this plate and dip these in different sets, which can be buffered and loading and different things which I'll explain you on the top. So once this is done, we can close this and the whole thing happens in this particular part. Now, if we see here on the screen, then we can see here this is the plate, how it looks, and we can define for the plate where the samples are there. So these are for the buffers, loading, and then buffers. So basically the idea is that first of all, we want them to, so if we can go to the slide, it will be easier.
Yeah, Shalini, I think we have to stop here. So thanks for showing the instrument and setup. It gives them idea that we are looking at antigen antibody reaction in much more high throughput manner, and that's where different type of principles are involved. Sometime looking at biolayer interferometry principle, or sometime looking at the surface plasmon resonance based principle. Just to summarize today's session, we have tried to give you glimpses. Of course, it is not a lectured session. We directly, with good coordination from all the TLs, which I should thank all of them for showing the things in a very short time, we were able to give you some glimpses at how these technologies at the time of pandemic were able to help us to understand this virus and the host response much better. And these technologies can be very powerful for various type of biological question which you would like to address in your journey towards your future of your research, right? So we got several these type of patents and products out from this research. And in the course which you are going to study in bioengineering and in the interactomics, you will be exposed for these technologies at much more length with the principal as well as hands-on session. Please do make this as an opportunity to interact with us much more so that we are there to help you, guide you through the processes. And of course, we are going to be having more of these kind of laboratory-based live sessions every month. And the next live session will be in the February. I did not see any technical queries on the forum from the student, but I definitely encourage you to talk to us more. Some other questions which were on the forum, essentially that in which way this, the technique which you're going to study in bioengineering can be helpful to you, especially for some students from the engineering background. Main idea is to expose you to a lot of concept and technologies at the early stage of your career, so that when you are trying to choose and build your career, you might find that, okay, biomedical engineering or interface of engineering to some domain of biology could lead you to some novel ideas and new concepts and maybe a new type of career journey, right? So that's where bioengineering course could be very powerful. Interactomics course is a much more specialized course to give you much more connection with the industries and applications. If you are to graduate and you want a job in the pharmaceutical sector, if you can spell out mass spectrometers and if you can spell out SPR and BLI very well with the principle and with at least basic data analysis workflow, you will be highly employable because a lot of industries and pharma sectors are looking at people having skills in these areas, right? So with this, I think, all of you should be really, motivated to work with us, given that there are thousands of students who are taking these courses. There is a huge number, but it still will be as much as possible to make it give you the classroom experience as much possible. Please do connect with us again next month for the laboratory-based live session. But additionally, you will have every week 2 live sessions on Friday and Saturday. With this, let me close and let me thank all the TAs for very well coordinating the entire session to give you the glimpses of how omics technologies and recent innovations in multi-omics has been so powerful and could be utilized for different type of biological questions to be addressed. Of course, today we are only focusing on the COVID-19, given that it has been, everybody is very much fed up with the COVID-19 and you'd like to see what, biology can deliver in this particular area. So we have focused on this concept. Nevertheless, these technologies are equally applicable, whether you work on plants or fungus or any kind of medical problem, these are going to be equally powerful to all of you. So we are almost now over the time and we would like to stop the session. So good luck everyone for continuing these MOOC courses. And we are very much optimistic that you are going to make the best use of these opportunities and courses to build your career further and take more knowledge from these courses. Thank you very much, and we'll connect again with you in the next live session in February. Good luck and good evening. Bye.